before your time to talk. And Hana, you're first, so we will introduce you after the intro.
Hi, Michelle. Just saw you on there.
Hi, Michelle. I didn't see you.
All right, well, I think we can get started. The first little bit of the session today is a repeat, so I'm sure some of our attendees from yesterday are going to join a bit late because it will be some repeated material. All right, good afternoon, everyone. Thanks so much for joining us today for this symposium, which is a joint event between PRiME at the University of Toronto and UCSF. I'm Shana Kelley. I'm the director of PRiME, which is an initiative focused on next generation precision medicine. And again, I'm really glad you decided to join us for this event. Just a few housekeeping notes on accessibility. We will be recording and posting this event so that it's as accessible as possible to people. Live captioning is enabled, so if you need it, just hit the live transcript function and you'll be able to see subtitles and be able to read what people are saying. The chat function has been disabled because of the size of the group that we're anticipating today, but the Q&A box will be enabled. The way that we'll be running today's session is that we're going to have 4 back-to-back presentations and then Q&A and discussion at the end. So please put questions into the Q&A interface as you have them. You don't need to wait till the end. We would love to hear from you. We've got a nice long block at the end for discussion, so please send us your comments and questions that you would like answered. If you have any issues during the symposium, you can get in touch with Angela from our team, and her email address is right at the bottom of the slide, and I'll give you a minute to jot it down if you want to have that handy. Okay. So then this is day 2 of this event. We had a fantastic session yesterday where we kicked things off with a set of speakers who focused in on the chemical and structural biology of proteostasis. And it was great science, great discussion, a really wonderful way to get this effort kicked off. And so today we will be turning to a different topic where there's lots of great work both at UCSF and also at UofT on the development of high-throughput technologies to accelerate biology. And so we'll come back to the schedule of the speakers in a few minutes. I do want to extend a welcome both from myself and also from Michelle Arkin, who is our co-organizer at UCSF. Michelle, give us a wave there. Michelle's a professor at UCSF, also the chair of the pharmaceutical chemistry department, and we'll hear from her in a couple minutes just with a short overview of the group there at UCSF. So I'm going to start with just a short overview of PRiME, just to give those of you who may be outside of Toronto or hearing about the initiative for the first time, a sense of who we are, what we're trying to do, some of our activities thus far. And then I want to tell you about the Global Challenge Program that is being funded at PRiME by CNOT, the CNOT Fund. And so we're very fortunate to have some resources to put to work on collaborations that arise out of the interaction between UofT, UCSF, and a few other institutions that I'll tell you about in a few minutes. But I'll just start off with a few facts and figures about Toronto. So I think of Toronto now that I've been here over 10 years, really as an epicenter for biomedical research. UofT is a large research intensive institution. We have over 90,000 students. We have 14,000 faculty. And in the biomedical research area alone, there's over $1 billion spent on research every year. Another really notable feature of our ecosystem is the fact that we have University of Toronto in downtown Toronto. We also have 2 suburban campuses, but adjacent to our downtown campus, we have a variety of research intensive hospitals. And so this creates a large ecosystem, a large community of people focused on biomedical breakthroughs. And so PRiME was put together, it was really two motivations. There was a top-down motivation at the University of Toronto to really bring people together, united by shared research interests. Since we're such a large university, it's easy to maybe not bump into a colleague for a while. But by having PRiME in place, we bring everybody together who's interested in disease biology, drug discovery, diagnostic development, and all the other areas. The bottom-up desire to do this was really along the same lines, in that we wanted a forum and a community of people that maybe don't see each other a lot because of we're in different departments, but by having a group that really unites everybody interested in these research areas, we thought we could be more collaborative and really accelerate what we're trying to do. So PRiME is what we call a strategic initiative at the University of Toronto. It's not a department, it's not a formalized group. It functions as a virtual research institute. And so what we've done is we have gathered up 75 faculty members from 4 different faculties within the University of Toronto: medicine, pharmacy, arts and science, applied science, and engineering, and canvassed these researchers from all of these different faculties, 12 different departments, to come together to be a part of PRiME. Every faculty member that's involved with PRiME brings with them 2 trainees. And so we have over 125 trainees, graduate students, postdoctoral fellows, who are also a part of PRiME. And this is a really impressive group of folks, very collaborative, translational, entrepreneurial, and it's really been exciting to see this emerge. We've been off the ground for about 1.5 years. Our goals at PRiME are to really elevate research in drug discovery, diagnostics, and disease biology at the University of Toronto, help our faculty members find resources for their research, really help them to accelerate their efforts. We also try to empower collaborations. We believe that collaborative research is a really powerful vehicle for new breakthroughs in the biomedical sciences. We also have a training mandate and spend quite a bit of time on trying to train the next generation of multidisciplinary and translational leaders. We also work to create community. As I said, there's a large group of people with shared interests, and we try to help bring them together on a regular basis. And we're also very passionate about building a biotech ecosystem, both at the level of helping science advance so that new startups can be created, also working with large existing companies in the pharma industry to try to really heighten the level of activity in all types of industry within Toronto. Our research strengths, so we're really on the basic research side of precision medicine, things like antibody engineering, all different types of high throughput omics, nanomedicine, diagnostic development. We have quite a few members of the chemistry department involved in PRiME, so there's a lot of work on targeted small molecules, biology on a chip systems, high throughput screening platforms, and as well, we have lots of people working on very interesting potential therapeutic targets. One of our main programs, and this is where most of the funding that we receive from the University of Toronto goes, is into this fellows program. So these are fellowships that we award every year to individuals working on projects that represent new collaborations between multiple faculty members that are a part of PRiME. We look for high risk, high impact projects to seed. And so then the fellow gets a year's worth of funding, full freight, to work away on a new collaboration. And then within PRiME, we try to scope a full research proposal around that project and then help the investigators get it funded. And this is a program that has been quite popular and successful so far. And we're now developing different tracks of other types of fellows programs. Another example of the types of activities that we pursue include a task force on COVID-19 that we put together back in March when the pandemic was first taking off. I think one of the very powerful things about this type of self-organization is that you already have a way to very efficiently put together the right group of people with interest in therapeutic development, diagnostic development, when a global crisis like a viral pandemic emerges. So now I'll turn to just giving you a little bit of background about the Connaught Global Challenge Program. So the Connaught Fund is a really tremendous resource that we have at the University of Toronto. This is a pot of money that was established in 1972 from the sale of the Connaught Medical Research Labs. And you may not have heard about these labs specifically, but you probably can recognize some of the discoveries that were made there. The labs were first put in place in 1914 to produce a diphtheria antitoxin. And then in 1921, when Banting and Best first discovered insulin, this is where the discovery was kind of expanded on, and these labs began producing insulin as well as other products like vaccines and antitoxins. So the Connaught Fund was then established at the University of Toronto and has been used over the last few decades to focus on or to support commercialization related research, but also to help organizations within the University of Toronto really establish a global reach. And so that's what we're trying to do through our involvement in this program. And so we were successful with a proposal that we put in to this program last year. And what we proposed to do was to try to leverage the research strengths that we have within PRiME, link with other leading institutions at a global scale. And what we ended up doing was to pick 4 universities across the world to partner with, UCSF being one, also Hebrew University, the ETH, and also NUS in Singapore. And so this proposal went in pre-pandemic. And so what it envisioned was a lot of people flying back and forth between these institutions, obviously a type of activity that we can't partake in right now. But once we do have our mobility back, we will be establishing a graduate student and postdoctoral fellow exchange program to really help new collaborations get off the ground between investigators at these different institutions. We're going to have a Frontiers of Precision Medicine speaker series, also a scholars program for undergraduate students. And this is just the beginning. This is just a way for us to get linked up. And then there are other mechanisms that we can use to continue building ways to collaborate and to do joint projects. So given that we're all confined to for the most part, to our home offices at the moment and our computers, we decided that what we would do is to kick off this effort with a series of joint symposia. So this is the first one that we're doing. And then we'll have subsequent events with the ETH, Hebrew U, NUS, and then we'll probably come back around and then start mixing and matching people from these different institutions to keep the conversation going. So we had, as I mentioned, a great first day yesterday where we got things started, and I anticipate we will have a great day today as well. Before we get going, I just wanted to give Michelle a chance to just talk to you a little bit about UCSF. Over to you, Michelle.
Great, thank you, Shana. So it's a big pleasure to be working with Shana on this. We went to graduate school together, and so it's always fun to reconnect. So just a very quick overview of UCSF, just to give you a sense of scale. It's public university, part of the University of California system, and medical center. Medical center reaches throughout Northern California, several hospitals. We have been the top public recipient of NIH funding for several years. And one thing that really distinguishes UCSF, it's completely devoted to biomedical research and biology research. And it has a terrific culture of collaboration and innovation and entrepreneurship. Almost 200 companies have started from UCSF investigators. We have about 3,400 faculty, about 25,000 staff. That includes 3,200 or so postdoc students and fellows, about equally divided. We don't have any undergraduates. It's purely professional school and graduate school. We have 4 professional schools: medicine, which is the largest, pharmacy, nursing, and dentistry, and 19 PhD programs in biology and biomedical sciences. So to help us organize this large institution that's really focused around, I mean, it's broad in biology, but it's narrow in the total spectrum of things that humans do in research. We organize ourselves in organized research units to help facilitate interdisciplinary research. And I just want to call your attention to the Institute for Quantitative Biosciences, QBI, as one of these PRiME-like ORUs that hosts a number of symposia with different institutions and stimulates collaboration. And it's the same, all of us are QBI members, I think, who have been talking from UCSF at this symposium. And that's another way to get into the UCSF ecosystem. There are so many people at UCSF in Toronto doing the kinds of research that we're talking about as part of our involvement in PRiME. We're just scratching the surface here. So if you see anybody at UCSF, come across a name and you'd like an introduction, please drop one of us a line and we'll be happy to introduce you. And I think with that, Shana, I'll hand it back to you for the symposium.
Great, thank you, Michelle. Okay, so the speakers that we're going to hear from today, I think all have really exciting, interesting systems that they have pioneered to help us to look at new aspects of biology. And so our first speaker today, and again, I want to remind you that we're going to hold the Q&A until the end of the session. So we'll just have rapid fire, one speaker after another. But then we have a nice long block for Q&A and general discussion. And feel free to type your questions as we go into the Q&A interface. Okay, so we are going to kick things off today with Hana El-Samad, and it's my pleasure to introduce her. She's the co-family endowed professor and vice chair in the Department of Biochemistry and Biophysics at UCSF. She's also a member of the Institute for Quantitative Biosciences. She was a Packard Fellow. She's won many other awards. She was named a Paul Allen Distinguished Investigator, and she's a senior investigator in the Chan Zuckerberg BioHub. She has degrees from Iowa State, also UCSB, and her research group really focuses in on the role of control theory and dynamical systems in the study of biological networks. So I'm really appreciative that you would join us today, and I'll turn things over to you.
So I'm going to start sharing now. Let's see if I can. Okay, let me put it in presentation mode. Okay, here we go. Good morning, everybody. Thank you so much, Shana, and thank you, Michelle, for the invitation. So as Shana just said, I'm actually not a biologist. I'm an engineer by training. I came into biology maybe a couple of decades ago because I was like deeply fascinated by how biological systems work. And specifically, I was very intrigued by the idea that the existence and persistence of biology proceeds in large part through the use of feedback control, which is the ability of a system to continuously sense its internal state and the world around it and react in real time with corrective action. So feedback control of this kind, as you might appreciate, is present at really every scale of biological organization, within cells, among cells, and throughout, the body, among organs, and all the way to ecosystems, as a matter of fact. Of course, this is something that students of nature have known for a very long time. So observations of feedback loops in physiology date back a couple of centuries. And just as a fun fact, actually the first cellular feedback loop was documented in 1956, way before actually RNA was known as a molecule. And it was isoleucine regulation in E. coli through what is now known as end product inhibition. So we know that feedback is very important for life to exist and persist. And on this slide is one of my really favorite quotes about that, and I want to read it. So beyond Earth, life without DNA is just about thinkable. One can imagine alternative strategy for storing information. Life without feedback loops, I have never met any biologist who can imagine that. So in my lab, we try to understand biological feedback systems by studying them using quantitative techniques. We think about how they fail and the repercussions of these failures in health and disease. We think about how we can fix such inherently dynamical processes such as feedback. And most recently, we started thinking about how to build feedback control system, feedback loops in cells and among cells in an attempt to bring that view and that capability of robustness to new therapeutic modalities such as live cell therapies. So today I'm going to talk about the engineering part. So I'm going to show you one of these feedback loops, and then I'm going to loop back, and that's pun intended, at the end to reflect a little bit on, since this session is about actually high throughput technologies, about the idea that we are sorely lacking those in studying such fundamental process of life. Okay, so this work is in collaboration with David Baker and his crew, Bobby and Scott at the University of Washington, Seattle, and the work is done by a fantastic graduate student in the lab, I should say previous graduate student in the lab, Andrew, who is now an independent fellow at our newly established Cell Design Institute at UCSF. So some of you may know David. David is, of course, one of the pioneers of understanding how proteins fold, and most recently in creating proteins de novo, designing them de novo, to do new functions. So these are the proteins that David really likes to think about these days are not variations of what exists in nature, but rather new forms. And my interest in this, and this is why I'm telling you this today, is the idea of whether we can actually use these de novo design proteins to build circuits in cells, so use them for cell engineering or synthetic biology. And the reason I like these designer proteins is that because they're actually simple. They're much simpler than evolved proteins, and as far as synthetic biology and cell engineering is concerned, this is actually turned out to be quite a property, not a flaw. So they're unifunctional. If they're designed correctly and the design is successful, they do one thing and do it precisely. I mean, of course, you compare that to evolved proteins, which are often multifunctional. They crosstalk with the rest of the cell because that's how they evolved to do. And you can start seeing how the attraction of these design proteins. So one such design protein is LOCKR, which stands for latching orthogonal cage key protein. So I'm not going to go into, I'll explain what it is. I'm not going to go into all the data that went into the design and the characterization, the biochemistry and the functional, studies, because this is all published. So suffice it to say that LOCKR is actually a bundle of, it's a de novo design protein. Nothing like that exists in nature. It's a bundle of 5 helices that are held together through a designed hydrogen bonding network, which of course David's lab has pioneered the design process. So I call that bundle a switch. And on that switch is a sixth helix, which is this thing, that I will call a latch. It is another helix that is mutated a little bit so that it is destabilized, so that it can swing open, okay? Threaded on the sixth helix, this latch, is a peptide that is used to functionalize this protein. And we'll get to that in 1 second. Now, this latch is opened through also a designer, a de novo designed key. So it's a peptide that can displace that latch and swing open the switch. So from now on, I will refer, this blue thing will be the switch. This red thing is the key. The key opens the switch, and this red thing here is that peptide that is threaded onto the switch, the functionalization peptide. Now you can imagine that you can thread any peptide really you want. So that bioactive peptide can be anything. For the purposes of today, that peptide will be, I'll talk only about one functionality. That peptide is a degradation peptide. So it's actually a CODC degron that is taken from mammalian cells. So basically what happens is that when the switch is closed, that switch and any cargo that is fused to it are stable because the degradation, the degron is hidden within the protein. Once it swings open, that degradation sequence is revealed, and then the proteasome can come and degrade that switch and anything really that is fused to it. This is a modular plug and play strategy. The cargo that could be on the switch could be anything. It could be a transcription factor, it could be an enzyme, it could be a kinase, and all of it will be recognized by the proteasome when the switch opens. And you can start imagining how that can interface with endogenous biology. Let's say you want to interrupt a signal in a pathway at node B, then all you need to do is fuse B to the degron switch. Of course, there are many ways to degrade proteins in cells. For example, there are auxin systems and so on. The reason I really like the degron locker is that because the key itself is not a small molecule, it's actually a genetically encoded peptide, then there is a possibility of starting to build circuits now. So both parts of that device can be produced within a cell. So of course, the first thing, given my introduction, the first thing we wanted to do with this is actually build a feedback loop. And it doesn't take much imagination to actually, think about how to do that. If you want to, let's say, induce, you have a linear pathway and you want to induce feedback on node B, then you just append the switch to B, and then you produce the key out of the output of this pathway. And that feedback loop can move anywhere from any node to any node, right? So that's a negative feedback loop, so that's easy. Now, does it work? So let me give you two very simple instances of how it works. Since actually the reason we approached this as part of a DARPA project was to build therapeutic cells, we wanted to, our first application to be a context in which we try to induce feedback on a pathway that kind of resemble a therapeutic production circuit, and see if by inducing this feedback on the circuit, we can buffer that therapeutic cell against disturbances. So fulfill really the promise of using feedback control. So the circuit is here. It's two transcription factors put in series with each other. So one activates its transcriptional cascade, and the output could be that drug that you want to produce within that therapeutic cell. The details really don't matter. So I'm going to refer to them as A and B, and A is one transcription factor. The other one is, the other transcription factor is B. And both of these transcription factors can be actually induced by small molecules. In this case, they're hormones. So these are inducible transcription factors. And the experiment I'm going to do, and this is to cut a very long story short, the experiment I'm going to do is I'm going to induce that pathway and wait until it settles certain output. So the cell is producing a certain amount of therapeutic output, and then I'm going to shake it. So for example, through that transcription factor B experiencing an increase of activity because something else within the cell caused it to do so, which is not an implausible situation to happen. And then I'm going to measure the output. So what happens when you disturb a system that has or doesn't have this induced feedback? So let's start with a system that doesn't have the feedback circuit. This is what we did. We induced the system, waited for a couple of hours until it settled on some value of the output, and then we hit it with a disturbance by increasing the activity of transcription factor B. And of course, as you imagine, that system is helpless to its environment. So the more you give a disturbance, the more it goes up to a new steady state, and you're producing more and more and more of the drugs. There is no remedy. The circuit has no remedy to its environment. Now let's add the feedback device. So I'm going to append A to the first transcription factor. I'm going to append the switch to first transcription factor. I'm going to use the output to produce the key as I just described, and I'm going to repeat this experiment. And this is what we get, right? What happened here is the known and loved homeostasis that cells do, and it's now induced artificially in a synthetic context. So basically what happens is that when the disturbance hits the system, the output increases, the amount of key increases, the amount of the switch that is active increases, and therefore that the transcription factor A gets degraded. So and this is when this starts decreasing. This is when we start compensating for an increase of activity in B and a decrease of concentrations of both A and B. And of course, as the whole motivation of this is to make cell therapies, transform them from a measure of last resort to an actual safe therapeutic modality, so that when things go bad, these cells have the ability to sense what's going on, correct for it in real time, and then go back to doing their thing once these disturbances disappear. But it is also very useful, and this is when I'm going to loop back, and I have only a couple more slides left. So I'm going to loop back to this idea of feedback and why we should care about it, even in a broader context than engineered cells. So as I said, this device is really modular. So what we did is we tried to actually test this concept in an endogenous context. And this context here is the mating pathway in yeast. So what we did is we fused the switch to sterol 12, which is the main transcription factor of this pathway. And then we produced the key in a very now hopefully similar kind of fashion to what we talked about before, to an output of the mating pathways through the mating responsive promoters. And we asked, can we actually modulate the output of this pathway in dynamic ways? And this is the data. And this is data, so this is the endogenous system that has no modification to it with all of its complexity. This pathway has many, many feedback loops. So it was actually not short of a miracle that we could, move it around in such a way, because it has its own corrective mechanisms. And these are different devices that we put in there. Some of them have keys that are designed to have lower affinity to the switch than others, and some of them have different promoters that are driving this key. So these are different kind of quantitative feedback parameters. So this is great, and this is me looping back. So this is great. What have we learned from all of this exercise? First, we demonstrated for perhaps the first time that the de novo designer protein can plug into endogenous biology and work with it rather seamlessly. These actually were not, I have to be honest, these were not difficult experiments. So we are actually, building increasingly complex circuits with these proteins, with way more than one component, and hopefully I'll talk about that with many of you soon. And I think this has implications for synthetic biology. Once we start building, combinatorially and reliably all of these functions, I think that opens up therapeutic spaces, as I just mentioned. But the lesson that I personally learned through this is that a little gizmo, I mean, look at this. This is a little gizmo. It's a switch that opens and closes and degrade, and degradation ensues. But once you add this gizmo and feedback, you can change dramatically a signaling pathway in ways that are not quantitatively predictable. I have, again, to be very honest, I actually do not understand quantitatively why these dynamic responses are so different. So let me so let me just leave you with a big message here that I hope I convey clearly. So I hammered for the last 20 minutes or so that feedback is actually used throughout life to give a system robustness to its circumstances. And that is true. What is also true is that there's no free lunch. So by adding feedback to a system, you've added a new vulnerability, and this vulnerability is the feedback itself. So what these data has shown us is that small changes, small differences in the feedback itself can cause big phenotypic differences. So this is actually a classical notion in control theory. You design your feedback loop, your feedback controller with the precision components. You pay a lot of money for that because you know the moment these are disturbed, even in the slightest ways, you're in for a world of trouble. So how much do we understand about that in biology, given there are thousands of these feedback loops in even the simplest life forms? How many do we really understand? How many can we predict dynamically and understand quantitatively? And do we understand at all what happens in disease? And can many disease be just a slight perturbation to systems that have evolved to protect you against perturbations and everything else except themselves? Okay, so that's, and we need methods. We really do need methods to understand all of that. So finally, I would like to finish by thanking the people who did the work. So Andrew, Galen, and Taylor, Andrew did most of the work. Galen and Taylor helped him with that. David, who is a fantastic collaborator, Scott and Bobby. And I actually wanted to talk to you about a new computational approach that we have to screen for these feedback dynamics. And I just left it out there just in case somebody is curious about that and wants to help us figure out the experimental platform to do that. So just to kind of tease your imagination a little bit. And of course, all our funding sources. And thank you very much for your attention. I hope I'm not way over my time. I lost track of time. I actually had a lot of fun talking about this today. Okay, so I'm going to stop sharing and I'll take my questions at the end of the session.
Great. No, you're right on time. Thank you so much. Okay, so we will move on to the next talk now, but again, jot down your questions, put them in the Q&A, and we'll have a great discussion at the end. And so it's now my pleasure to introduce my colleague, Jason Moffat. Jason's been with us at U of T since around 2007. He is an expert in functional genomics. He's established the platform for advanced cell engineering, which is really just an amazing enterprise that has produced a lot of different projects, many different publications since its inception. Jason is very well known for creating the Toronto Knockout CRISPR guide RNA libraries that consist of over 90,000 guide RNAs. And they've deployed this type of whole genome CRISPR-Cas9 screening to a variety of really interesting problems. And I think that is what Jason is going to talk to us about today. I just want to mention that he's also the founder of several biotech companies, including Northern Biologics, Pioneer, also Empirica, and he's a senior fellow of the Canadian Institute for Advanced Research and a Canada Research Chair. So with that, I'll turn it over to you, Jason.
Okay, thank you very much for that introduction, Shana. Can everyone hear me? I hope.
Yes.
Okay, great. So, first, I'd just like to start by thanking Shana and Michelle and all the organizers of this symposium. It's been very interesting, and I appreciate the opportunity to talk about some of the work we're doing in my lab. So, let me get this straight. Okay, so mostly our lab really thinks about the genotype to phenotype problem, and mostly in human or mammalian cells. And so we build a lot of genome-wide loss of function libraries to assay the effects of systematic perturbation on various phenotypes. And our favorite phenotype really to study is cell fitness. It's a highly sensitive phenotype, and it really represents an underlying highly connected network of genetic pathways that communicate to regulate cell growth and proliferation. And so, well, we've been doing this for some time now using different technologies like RNA interference and more recently CRISPR. And at one point we conceptualized this model that I'm showing on the left-hand side here, we called the DAISY model of gene essentiality, where we think that there's probably a core set of genes that are important for cell fitness that don't really change across most cell types. And it wasn't until we had CRISPR technology that we could actually quantify these core fitness genes across a number of different measurable properties that are shown here on the right-hand side of the screen. So things like correlation to gene expression and variation in gene expression, as well as protein interactions. So I just wanted to make note, and thank you, Shana, for introducing this, but we make all of the libraries that we generate available on Addgene. So our first attempt at making a CRISPR library, we called it Toronto Knockout version 1 library, or TKL version 1. We learned a lot from that library, and that allowed us to reduce the size and make each of the guides more efficient. We're now up to version 3 of the library, and we've been using that for the last few years. And that's been really useful for the community here in Toronto and abroad. So today I'm going to talk about cancer intrinsic immune evasion and how we started to map genes important for cancer intrinsic evasion to cytotoxic T cell killing. Now, before I do that, I'm just going to make a note here that I think we're at an incredible juncture in genetics for mammalian cells, and especially genome-wide screens. This is something that when I started doing my PhD over 2 decades ago now in yeast, we couldn't even conceive of doing this in a human cell line or in a mammalian cell line. And now, over the past 5 years, really since CRISPR has emerged, my last count, there are probably about 1,000 different human cell lines that have undergone some form of genome-wide screen. And this is just completely astounding because it really starts to let us link together genotype to phenotype relationships really quite efficiently. And so I think the best outcome so far of all of these screens is that we can derive a list of core essential genes that can be used to improve our efficiency at annotating the functional genome for human genes. And so I'm just going to make a quick mention too that a few years ago, once single gene perturbation libraries started coming online, lots of groups started to attempt to make multi-perturbation libraries, and the field is kind of a subfield of genetic screening in the CRISPR field, but it's come quite far. I just wanted to plug a library that we recently, or a system we recently published on, which was a really nice collaboration with the Blanco lab, where Thomas from his lab and Michael from my lab teamed up with Kevin from my lab to generate this thing we call Chimera, where we can fuse Cas9 and either a single or multiple Cas12a guides together and have a very efficient multiplexed editing system. So if you're interested in that, there's a bunch of detail in the paper. So many people have probably seen a plot like this. This is a survival plot showing survival of patients treated with immune checkpoint therapy. We became interested in this many years ago because of this extremely long durable tail. And there are two things that are really interesting from a functional genetic screening perspective for us. One is that, not all patients respond to these drugs, which suggests that there's a lot of non-responders that may not have the genetic circuitry wired up in their tumors to be able to respond to these drugs. And then the other issue is that many of these patients may just undergo some sort of de novo resistance or adaptive resistance to these therapies. So we started thinking about how to set up screens that would allow us to probe resistance and also sensitivity to cytotoxic T cell killing. And this project was really led by a former PhD student who's a surgeon scientist in training, Keith Lawson. And so we focused heavily on one aspect of the cancer immunity cycle, and it's this aspect here where T cells are activated and are targeting tumor cells by various mechanisms of cell death. And so if the immune system worked perfectly to protect us from cancer, then the model would work as follows, right? So cancer cells release these antigens that are recognized and presented by dendritic cells to naive T cells in the lymphatic system. And then naive T cells that recognize tumor antigens would evolve into these cytotoxic T cells that circulate back to the tumor and eradicate tumor cells through an arsenal of different cell death mechanisms. So what we did was we asked this question: can we find cancer cell intrinsic genes that regulate cytotoxic T cell killing that are broadly conserved across multiple different cancer models? So in order to do this, what we did was we took advantage of naturally occurring mouse tumors, and we found 6 different mouse tumor cell lines, 2 colon, 2 breast, 1 kidney, and 1 melanoma. And we engineered those cells to express a surrogate antigen that's recognized by a transgenic T cell that we can isolate from spleens of the corresponding proper genotype of the mouse. So there are these different tumors are actually derived from either BALB/C or C57 black 6 mice. So you need different transgenic T cells to kill them off. The tumor antigen in the BALB/C background we used was hemagglutinin. The one in the black 6 background was ovalbumin. So we also created a mouse library based on all our best knowledge of how to design guides targeting the entire mouse protein coding genome. And what we did was we introduced this library into each of these different 6 models and created reference knockout populations. And from these populations, what you can do is we split the population into 2 different arms, And we did a dropout screen to look for genes that are essential for growth of each of these cancer cell lines. And we also did a screen where we treated these cancer cells with these activated T cells that are targeting specific tumor antigens we express on the surface. So from these screens, what we can do is we can get essential genes and identify the core essential genes across each of these, well, across the 6 models that we're using. We also get sensitizer or negative interactions for T cell killing, and also suppressor or resistance genes or interactions for T cell killing. And then we can generate this other sort of DAISY model of genes or CTL evasion genes. This is just a quick note to show you that context dependent essential genes or fitness genes are actually quite prevalent across the 6 models we use. So we're actually studying biology from different models that rely on different different pathways. So we're not studying the same genetic wiring in each of the 6 models that we've used. And so the results of the first T cell killing screen are shown here, and these were in Renka cells. And this is shown as a rank plot, with each dot being a gene targeted in our screen, stratified by what we call a normalized Z-score. And this quantifies the relative effect of knocking out that gene, followed by a growth in the presence or absence of T cells. So what was interesting when looking through these lists, so the yellow dots here are suppressors or resistors, and the blue dots down here are sensitizers or negative interactions. So what was exciting when we were looking through these lists was that we had a good idea the screens actually worked, because we found top resistance hits across many of the known pathways that were already implicated in resistance to T cell killing. For example, loss of interferon gamma receptors, loss of TNF receptors, JAK, STAT signaling. Also, you can see in here is B2M, TAP2, and this should say TAP1. These are genes important for antigen presentation. Moreover, when we looked at the sensitizing hits, we found known immunotherapy drug targets like PD-L1 down here, which provided further support that the screens were working. So we went ahead and we did the other 5 cell lines, and we aggregated a lot of that data into this map here, where we filtered essentially gene lists whose pathways emerged in more than 3 out of the 6 screens. And what you can see is that a lot of the sort of known resistance and sensitizing pathways and innate immunity, JAK, STAT signaling came up. So we were encouraged by that. But we also identified other pathways that looked interesting and novel, or at least less studied in the context of CTL evasion. So we wanted to really zoom in on what those specific pathways were. We wanted to understand this core cancer intrinsic CTL evasion gene set. So we narrowed our target genes to those that scored with an FDR of less than 5% in 3 or more screens. We came up with this list of 182 genes. And we created a small validation library, or we called it the mouse validation library. And we repeated all of the screens again with that library. And this basically confirmed most of our primary screening results. And the major pathways that are driving either sensitivity or resistance to T cell killing are shown here on the right. So the next question we had was, which of these 182 genes are really the most robust genes that mediate sensitivity or resistance to CTL killing? And so we rank ordered those genes. And this is displayed here on this volcano plot. And this identified actually many regulators of interferon gamma pathway, including genes that negatively regulate this pathway, and were recently identified, or not so recently, but a couple of years ago, identified in an in vivo synthetic lethal screen with immunotherapy by the Hana lab at the Dana-Farber. We also found genes in this pathway that relate, that mediate resistance too, right? So things like interferon gamma receptors, JAK, STAT. So we started really wondering if the interferon gamma response is a subphenotype of cytotoxic T cell killing, and we could somehow tease out all the genes that were important for interferon gamma sensitivity relative to CTL killing. So what we did was we performed a genome-wide interferon gamma sensitivity screen. And what we found is shown here on this plot. So again, the sensitizers are in blue and the resistors are in yellow up here. And so overall, a large number of the sensitizers overlapped with our core cancer intrinsic CTL evasion genes. And this confirmed that this cytokine was quite important for the overall CTL evasion phenotype. So I think we can think of interferon gamma sensitivity as a subphenotype, but I guess, and that's sort of shown here as a Venn diagram of the core CTL evasion genes intersected with the interferon sensitizers. And so perhaps the most interesting thing was the top hit, which was this gene called FITM2. So this is a gene whose knockout really sensitized these Renka cells, at least, to interferon gamma toxicity. And FITM2 plays a known role in lipid droplet formation, but has not previously been reported to have a function in the context of immune biology. So to better understand FITM2, what we do when we don't understand something is we usually make a knockout and do another screen. So we generated FITM2 knockout cells and in the Renka background, and we validated that the phenotypes we expected from knocking out FITM2, this lipid droplet regulator, were real. And then we did an interferon gamma sensitivity screen again. And we can basically then measure genetic interactions because we have FITM2 knockout and we have the parental or wild type results. So this is a busy slide. I'll walk you through it. But these, so what I'm depicting here are the results of the FITM2 sensitivity screen to interferon gamma relative to the parental or the wild type cells. So FITM2 is on the vertical axis, wild type is on the horizontal axis. And what you can see right away is the screen appeared to have worked quite well because you get this appearance of FITM2 itself as a positive hit. But what's really happening is because it's such a strong dropout in parental cells in the presence of interferon gamma, there's no effect at all in the FITM2 knockout cells because FITM2 is already knocked out. It appears as a positive, but it's really not. It's just a masking interaction. So you can see all kinds of other interesting stuff. For example, the emergence of proteostasis regulators, fatty acid synthesis regulators, autophagy and peroxisome. So these are all pathways and genes that seem to be interacting in a way with FITM2. So every gene on this plot you can think of as a double knockout with FITM2. So overall, this allowed us to build a model for how FITM2 works in the context of interferon gamma response. So we know that when FITM2, that when interferon gamma signaling occurs, there's a buildup of reactive oxygen species. And this can cause an accumulation of oxidized lipid and protein products resulting in cell death. So we think that by knocking out FITM2, these toxic lipid species can no longer be sequestered in lipid droplets, so making the cells more sensitive to lipotoxicity. We haven't proven this, but the genetics points to the possibility, I guess, that processes that increase lipid species such as peroxisome biogenesis and autophagy can buffer this phenotype when you knock them out. Whereas knockouts of processes that cause cells to experience more lipid or proteotoxicity, including accumulation of free fatty acids and protein waste down here, can exacerbate this phenotype. So we can take these results and in addition to the interferon gamma screens, we also perform CTL killing screens in FITM2 knockout cells. And then you can plot the results of both of those different screens on the same graph. And so we call these genetic interaction maps. And the idea here is that if genetic interactions are shared, then mechanistically, they should impact the overall phenotype, which is CTL killing in this case, with the interferon response. So this data is, we call these sunbeam plots. And this revealed sort of a striking finding that autophagy genes were no longer suppressor or resistor hits in the CTL killing screens. So that was kind of a surprise. And this suggested to us that these autophagy genes are probably having non-interferon mediated effects in these CTL killing assays. So what we did next was we took our data and we built a genetic co-similarity heat map, and we clustered all of that data together to identify shared dependency profiles. And so in this analysis, what we do is calculate correlations between all pairwise gene combinations for their similarity to CTL killing profiles across the panel of screens. We use all of our time point information. I didn't mention that at the beginning, but we usually sequence multiple time points. And the basic idea here is that genes that are correlated with each other share similar functions or mechanisms regulating the phenotype. And so what's interesting is that macroautophagy emerges a pathway theme on the heat map. And this allowed us to really zoom in and look for correlated genes on this. And what we found and what was clear was that genes involved in TNF signaling and NF-kappa beta signaling were prominent in this subnetwork. And this allowed us to hypothesize that maybe autophagy regulates TNF-mediated cell death in the context of these RANCA models. So we went on and we did a TNF sensitivity screen. This was a bit of a debate in the lab whether to do this screen. And it was really, I guess, motivated by reviewer comments to do it, because when we threw TNF on RANCA cells, we had no effect whatsoever. So we were debating whether or not to spend the money to do the screen, but eventually we did. And what you can see here is that in the presence of TNF, a lot of the autophagy genes became sensitizers to this phenotype. And so this really kind of validated in our eyes that autophagy was a major pathway that responded to TNF-mediated treatment. And so what we did to validate this was we used we did an independent experiment using TNF blocking antibodies, and we were able to demonstrate that in an ATG12 knockout, two different ATG12 knockout clones, they were sensitive to the presence of TNF, but when you added in TNF blocking antibodies, this rescued that effect. And so we went on again and we took ATG12 knockout cells. We did TNF sensitivity screens, and we also did CTL sensitivity screens, and we generated the same kind of data that we generated for FITM2 and interferon gamma. So on the left-hand side is TNF sensitivity in ATG12 knockout cells versus parental cells compared to CTL sensitivity in ATG12 knockout cells compared to parental cells. And I guess what's quite surprising from these findings were that additional perturbations in the autophagy pathway, so every single gene that's noted on every spot on here, you can think of as a double mutant with ATG12. So what's notable though is that additional perturbations in the autophagy pathway mediated resistance in this ATG12 knockout background. And we did them in different clones and even in cells expressing HA and not expressing HA. So we know this is not a clonal effect. So this was very unexpected. And it indicates that making a single change to the genetic background can sometimes lead to the opposite phenotypic effects. So it underscores the need to consider genetic interactions when devising ideas about therapeutic strategies. So the last thing I'll mention is that we wanted to, given the important role of autophagy in regulating CTL killing in vitro in cultured models, we wanted to further study its relevance in vivo. So in order to do this, what we did was we performed an additional screen with this mval library, this validation library targeting 182 genes. We did this in both. What we did was we took that library and generated a reference knockout population, and then we injected those cells either into mice containing an immune system, so they were immunocompetent mice, or mice that were impaired in their immune system, or NCG mice. And so when we compared the distribution of guides between the two mouse strains, we generated over 180 tumors in total. So we had lots of numbers. When we compared all the guides, we noticed that autophagy genes were strongly depleted and selected against in the immune competent, i.e. the BALB/c mice versus the NCG mice. So this told us that autophagy is an important cancer intrinsic immune evasion gene or pathway in vivo as well. So we're further exploring this in other models as well to see how common this is. Other labs have also identified autophagy as an important cancer intrinsic evasion pathway and validated. The Burso lab, for example, just published a paper validating that TNF was a mechanism that sensitized autophagy knockout in his pancreatic models. So it looks like maybe there are multiple models that where this biology is real. And so just to finish, so I've shown you a few different sort of vignettes. First bringing you through, I guess, one of the first studies to look at many different models of cancer and map out core cancer intrinsic CTL evasion genes, how you can take that information and move forward to try to really tease out the underlying biology for specific hits and specific modes of killing, for example, interferon gamma, and then look at genetic interactions and how those may play into either the target phenotype or subphenotype, what you think are subphenotypes of that target phenotype. And so this has recently been published if you want to get more details. And I think I'll just end by acknowledging everyone that participated in this project. So this was a really big team effort from the lab that was led mostly by Keith Lawson and two former master's students, Sally and Rumi, and with a lot of help from Seal and a PhD student in the lab, and Joost Komens, who's a postdoc in the lab. But everyone really pitched in here. It was a really nice collaboration with Agios Pharmaceuticals and Chris Souza, and a number of additional labs in Toronto, and some analysis help from Trevor Hurd, a former postdoc in my lab, who's now got his own lab at MD Anderson. So I'll end there and take your questions later. Thank you.
Great, incredible story, Jason. Thank you. All right, so we're going to keep moving. Again, pop those questions into the Q&A box. And our next speaker is Zev Gartner from UCSF. Dev is a professor in the Department of Pharmaceutical Chemistry. He got his PhD with David Liu at Harvard, was a postdoctoral fellow at Berkeley with Carolyn Bertozzi. He's received an NIH New Innovation Award or New Innovator Award and a DoD Era of Hope Scholars Award. He was named one of the Brilliant 10 by Popular Science, and he's a Chan Zuckerberg BioHub investigator. His lab looks at how cells self-organize into tissues and how the structure of tissues help regulate cell behaviors and how tissue structure breaks down in diseases like cancer. All right, take it away, Zev.
All right, thanks a lot, Shana, and also to Michelle for the cool opportunity to be able to participate in this symposium. So as Shana mentioned, my lab's really principally interested in learning how to build tissues. So we're interested in the structure of tissues, and Anyone who's ever looked at a sample of a tissue from a pathologist or at the structure of, say, organoids in a dish probably recognizes that although tissues have a certain average structure, there's actually considerable heterogeneity in the way that cells are organized, even in healthy tissues. And so in thinking about this and seeing this come up over and over again in our efforts to engineer tissues in the lab, we became really interested in fundamental processes that might be driving the balance between order and disorder in tissues, both engineered tissues and in vivo. And so we wanted to begin to investigate what was the source of these competing processes. We thought that this was a very important question to investigate more broadly because the balance between order and disorder have a really important set of consequences. For example, in development, if tissues properly organized, you get a baby out at the end of the process. And if something goes wrong, that's obviously a disaster. Similarly, during progression of diseases such as cancer, which undergo sort of stereotypical changes in structure and loss of structure as the disease progresses, clearly this balance between order and disorder is becoming disrupted. And in the context of our discussion today, if we're going to begin to use tissues like organoids or other engineered tissues for platforms for example, screening for how cells interact, as we saw in Jason Moffat's presentation, or for drug screens, for example, we're going to need to begin to understand what gives rise to the underlying heterogeneity that we see in these cultures, because this is going to complicate our interpretation of data. So two really brilliant scientists in the lab, Dr. Vasudha Srivastava and Jennifer Hu, decided to investigate this question. And we had a lot of help along the way from Matt Thompson and Mark Labarge, and we benefited greatly from discussions with Rob Phillips and Greg Huber. So Vasudha and Jennifer began by using the human mammary gland as a tissue. And this tissue is really great because it has a really well-defined average structure. So if you look across the ducts of the mammary gland, what you see on average is a structure shown here. So the tissue has at its center, lining the lumen, a population of cells called luminal epithelial cells in green. We call them LEPs for short. They're surrounded by another population of cells called myoepithelial cells, or MEPs, shown in magenta. And the entire tissue is encased in a specialized form of extracellular matrix called the basement membrane. And the balance between order and disorder is important here. The green cells secrete milk into the lumen, and the magenta cells contract to push the milk out. And so if this structure gets disrupted, even under normal conditions, that's going to mess up the function of the gland. More broadly, this basic organization is shared by a whole variety of other glandular tissues in the body. And also, this general scheme of a multilaminar tissue is one that occurs frequently in development and in the adult human body. So Vasudha and Jennifer began by putting together an assay for the self-organization of this tissue. It begins by taking reduction mammoplasty tissue that's donated by human patients. We can dissociate it into the LEPs and the MEPs, and then we can take these cells and transduce them with lentiviruses to either up or down regulate various proteins or pathways. We can then mix these cells back together at defined ratio to make reconstituted organoids of defined size and composition. And when we put these back into basement membrane gels like Matrigel, they begin to self-organize and sort themselves out, just as you see in this video here. So we wanted to begin to quantify the sort of heterogeneity in structures that happen in this assay. And so what you're looking at here is a snapshot at around 36 to 48 hours of what these tissues look like. What you can see is that some of these tissues, like this one here, are very well organized. Some of them less so. So we put together a image analysis pipeline that begins with a machine learning algorithm and essentially allows us to segment the various cell types in these tissues. And then what we can do is we can quantify the relative ability of these LEPs and MEPs to position themselves at the outside of the tissue by adding up the fraction of magenta and green tissues in these examples here. And so, for example, this tissue here is greater than 90% MEPs at the tissue edge. aggregating these data, you get these probability distributions like the one shown here. And so what you see on the x-axis here is the fraction of these LEPs at the tissue edge. So over here at 0, this is a perfectly organized tissue. Over here at the right is a perfectly inverted tissue. And what you can see is that on average, about 20 to 30%, the tissues have about 20 to 30% LEPs at the tissue boundary. So this was quite interesting to us. What it showed is that very clearly these tissues are biased towards an organization where you have more MEPs on the outside. So there's order there. But also the distribution is pretty broad, right? It's not a delta function over here at 0% LEPs. And so this led to sort of two questions. What was driving this order? Why do we see mostly MEPs on the outside? And similarly, what was driving the disorder? So why is this distribution so broad? So a number of years ago, we were able to shed some light on this first question. What we learned is that tissues like the mammary gland that have this multilaminar structure self-organize by trying to essentially minimize the mechanical surface energy between all the cell-cell interfaces in the tissue. So just to give you an intuition for how this works, if you have a green-green cell interface that's high tension and therefore high energy, it will tend to shrink, allowing lower tension and lower energy interfaces to expand to replace them. So we're actually able to go into these cells in the mammary gland and measure these relative interfacial energies, and we put together an assay to do this. The basic assay is we take two cells of various types, we allow them to come into contact and form an interface, and this interface achieves a steady state, which depends upon the interfacial tensions at either the cell-cell or the cell-medium interface. And so we can calculate the energy of these two configurations according to the equation shown here. So these gamma terms are going to be those interfacial tensions. We can measure the interfacial tension at the cell-medium interface using a technique called micropipette aspiration. We can then relate that to the interfacial tension at the cell-cell interface by Young's equation. And this requires us to measure this contact angle here, theta. I'm not going to walk you through the data here, but the interesting finding that came out of this analysis was that although there were quantitative differences in the interfacial tensions at the various cell-cell interfaces in this tissue, what was really dominating the self-organization were the differences in interfacial tension between the MEP ECM interface and the LEP ECM interface. So this was by far the most energetically favorable interaction, explaining why the MEPs were on the outside. And this was the least favorable interaction, explaining why the LEPs were excluded from the outside. So this explains why this distribution is shifted to the left. But we began to ask, how organized is this distribution anyways? So for tissues of the size and the composition that we are building, what would actually a random distribution look like? So to begin to answer this question, we put together a relatively simple experiment where we built tissues of the exact same size and composition, but now where rather than using MEPs and LEPs, we took a pure population of MEPs, split them into two pools, stained one green and one red, and mixed them back together exactly as we did before. When we image these tissues, we still see a lot of cell dynamics, but the cells do not appear to sort out, at least by eye. And when we quantify the distribution of structures that we see at the same time point as in the previous assay, we get the distribution shown here. So this is nicely centered at 50% green and red cells at the tissue edge, which is the exact composition of these tissues. And it's also very nicely fit by a Gaussian. So this is random in this tissue. So this raised a couple new questions, right? So we can conclude up to this point that the expected distribution that we would see in the absence of any directed energies based purely on probabilities would be this gray distribution here. If we looked at this question purely through the lens of interfacial mechanics, we would expect to see a distribution centered at 0 here, a delta function. So the question then becomes, why do we get this blue distribution? So what sets the balance between these two extremes? So an important clue came to us when we began to image many tissues dynamically. And what you can notice is that going back to the tissue that I described earlier, although it seems to have an average structure with about 90% MEPS on the outside, it seems to actually be sampling dynamically from many different configurations of the tissue over time. And so we can trace this out by plotting this structural coordinate along the y-axis here and time along the x-axis. And what hopefully you can see is that as we approach the steady state at around 36 to 48 hours, this tissue is dynamically and seemingly randomly sampling back and forth between these configurations. And we can look at more and more tissues this way. And what we conclude is that it appears that individual tissues at the steady state are sampling from the same distribution that the entire dish is sampling from at a single snapshot in time. And so what this appears to say is that this system is behaving ergodically. So one very nice way to model systems that have this property is using Boltzmann statistics. And so we can begin to reformulate this problem by describing the probability of observing the ith macrostate, which we define as one bin along this structural coordinate here, according to the expression shown on the right here. And so this has a number of different parameters. So one is this one which we call the configurational entropy. And so we can calculate this quantitatively by just measuring this grade distribution shown here. It's essentially the number of ways of putting together one of these macrostates. All right, so that's something that we can measure. We can also measure the energy of the ith macrostate shown here by multiplying the interfacial energy of each flavor of interface in the tissue times its area. So we can measure that. And what sets the balance between these two terms is this parameter here, activity, which you can think of as being analogous to a tissue temperature. And we think that this is best represented by the random component of cell motility in the tissue, or these sort of random mechanical impulses that the cells are applying to their surroundings. And this entire distribution is normalized using the partition function Z, which is a function of all these parameters. So this seemed like a really great framework to begin to understand this balance between order and disorder in the tissues. And so we wanted to begin to probe it somewhat more rigorously. And so we decided to do that through the lens of the structural changes that occurred during breast cancer progression. And so this diagram is meant to just give you a simple idea for what happens during breast cancer progression. So it emerges specifically within the luminal cell population through activation of oncogenes like PI3 kinase. This then allows these cells to proliferate and selectively expand where they fill the lumen. And then through what appears to be a stochastic and poorly understood process, these cells begin to push into the myoepithelial layer where they gain access to the basement membrane, and eventually they break it down and are able to invade into the stroma. And so oncologists are really trying to understand what happens during this process because there's a huge inflection of risk as you move to IDC. So we hypothesized, and sort of this has to occur if you think about it, there must be a intermediate structure along this trajectory that places those luminal cells in contact with the basement membrane. And so we wanted to begin to ask, how could this model help us understand what could contribute to this? So to further begin to frame this question, we turned to a really interesting mouse model of PI3 kinase driven breast cancer for inspiration. And so in this model, which is cited down here, you can activate PI3 kinase stochastically within the luminal compartment. And after several months, these mice will go on to form a whole variety of really heterogeneous tumors. But interestingly, the authors noticed that at early time points, around 4 weeks, that these cells, which were marked by fluorescent protein and express PI3 kinase, were observed to begin to populate the basal compartment of the tissue. And so this provided a really great platform to begin to investigate what gives rise to this intermediate. And so going back to our model, you can imagine several different ways of generating this intermediate. One is you can decrease the energy difference between having a LEP and a MEP next to the basement membrane, right? So I'll describe how that could happen. We can imagine increasing the fraction of LEPs in the tissue too. This is also going to shift this distribution to the right. And finally, you can imagine changing the activity in the tissue. So all of these changes are going to serve to shift the distribution of structures to the right, putting more LEPs in the basal compartment. And critically, the really cool thing about this model is that all of these really abstract terms can be boiled down to a specific measurable property in the cells, right? So in this case, contact angle, over here, the fraction of cells, and down here, the random components of cell motility. So we can show very nicely that increasing the fraction of LEPs in the tissue, all other things being equal, increases the composition of the basal compartment, shifts the distribution to the right. So these are wild type tissues with 25% LEPs, 50% LEPs, and 75% LEPs. So exactly as predicted. We can also hypothesize that in this PI3 kinase driven mouse model of breast cancer, that if the delta E's are changing in the system, this would correspond to an increase in the contact angle between these PI3 kinase expressing LEPs and their substrate, right? So that would be representative of a more favorable interaction with the substrate. To test this, we went back to our human model. We grew up our cells. We infected them with lentivirus expressing a GFP as well as an activated PI3 kinase. We reconstituted them back into tissues and measured this structural distribution, and then also measured the contact angle of the luminal epithelial cells with their substrate. So these are the contact angles for wild type luminal and myoepithelial cells. And this is the structural distribution, the fit for it for wild type tissues. When we put PI3 kinase into the LEPs, just as predicted, the contact angle goes up, shifts the distribution to the right. And as a rigorous test for whether it's the change in interfacial energy which is responsible for this change in structural distribution, we can go back to those PI3 kinase LEPs, knock down talin 1, which decreases the contact angle back to near wild type, and this shifts the distribution back to the left again, rescuing the structure. So finally, we wanted to investigate the role of activity in these tissues. So just as increased activity is predicted to disrupt tissue structure, you can also imagine a scenario where you still have an average structure which is more or less organized, such as I showed in the previous slide with PI3 kinase. In that scenario, you would predict that decreasing activity would actually rescue tissue structure, all other things being equal. So same composition and same contact angles. So we wanted to ask, could we identify perturbations which would change activity, which here we're going to use the radial component of cell velocity as a surrogate for activity. So perturbations which change activity without changing these contact angles and which rescue tissue structure. So going back to this assay, we basically did the same thing, added this cell velocity measurement. And we found one perturbation, inhibition of AKT using MK2206, which caused a marked decrease in the median radial velocity of these cells in the tissue. And remarkably, this shifted the distribution back to the left again, so exactly as predicted by the model. So to summarize, what we learned in this study is that it's really critical to think about the entropy of different configurations of a tissue in terms of trying to understand this balance between order and disorder. So not all tissue structures are equally probable. And what tips the balance, or what pushes tissues towards a structure of organization, at least in the context of the mammary gland, are these differences in interfacial energies. So this helps shift that distribution to the left. And activity is this key parameter that sets the balance between the influence of these interfacial energies and the influence of entropy in determining what types of structures you get at the steady state. And so you can imagine mapping out essentially phase diagrams for how ordered a tissue will be given these two parameters. We think that this framework for thinking about tissue self-organization provides a lot of insight into how breast cancer progresses. And you can imagine changing the tissue structures and these forces that are responsible for cell positioning in the context of other tissues to begin to think about changes in structure. And we also think that the same framework can be applied to thinking about development. So thank you all for your attention. And I just want to again point out Vasudha and Jennifer, who were phenomenal leaders of this project, all of our collaborators, and all of these organizations for their generous support.
All right, thanks a lot, Zev. That was great. Okay, so I'm the last speaker in the session, and then we'll move over to the Q&A and discussion. And while I share my screen, I'll just give myself a very brief intro. I already told you that I'm the director of PRiME, also a faculty member based in our Department of Pharmaceutical Sciences at the Faculty of Pharmacy, but also in our biochemistry department, chemistry, and also biomaterials and biomedical engineering. So I'm really pleased to be part of this session today. It's just incredibly exciting science of so many different flavors that we've heard about so far. What I'm going to do in this brief talk is just to tell you a bit about a platform that my group has developed and then really exploited in collaboration with some of the other investigators in PRiME to try to provide new systems to allow us to really accelerate what we can learn about biological systems. And the rationale here, it comes from the idea and the inspiration from the last several, almost a decade now of really powerful disease biology that has been elucidated, by single cell analysis systems. So certainly areas like neuroscience, immunology, stem cell biology, and certainly oncology have really benefited from the ability to be able to look at one cell at a time and to make really precise, highly accurate measurements using single cell profiling systems. This gives us the opportunity to really deconvolute heterogeneity, understand how different cell types are working together to produce more macroscale phenomena. And so again, there's a lot of powerful systems out there for single cell biology and a lot of breakthrough insights that have been gained through the application of these types of systems. But I would argue that where things get difficult is when you're trying to look at very large populations of cells. So hundreds of thousands, millions, billions of cells. Most existing single cell analysis technologies really require that you almost look at cells one by one and sort through them to try to find the interesting ones that are maybe the ones that have a phenotype that are what you really want to be able to get at. And this, of course, if you break it down into time, presents a problem. Bandwidth, it presents a problem, and it also limits the types of systems that we can look at with existing solutions for single cell analysis. So we've been working over the last few years to develop some new technologies here that are really focused on allowing us to look at really complex heterogeneous systems. So let's say human blood, where a milliliter contains billions and billions of cells, red cells, white cells, and many different cell subtypes. Other types of biological samples, tumors, or man-made heterogeneous samples like libraries of CRISPR-edited cells, or sorting through large populations of cells to, let's say, look for T cells that are tumor reactive. And so we've had an eye on trying to have a way to look, again, at collections of billions of cells to try to pull out individual cells, small collections of cells that may be doing something interesting phenotypically. So I'll take you through this platform that we now have up and running and have been working with for a few years. And then I'll tell you about how we are applying this to phenotypic screening using CRISPR-edited cells to be able to really enhance what we're able to do with looking for rare phenotypes and then really understanding their biology. So this platform, we refer to it as magnetic ranking cytometry. And it certainly is inspired by flow cytometry. So in flow cytometry, you take a fluorescently labeled antibody, you introduce that into your sample of cells, you pass the mixture of cells through a flow cytometer, and then you can measure how much protein expression you have on those cells by measuring the fluorescence signal. It works beautifully. It's a workhorse of many different subfields of biology. Where flow cytometry does not work really well is when you have a really heterogeneous sample, or if you have a rare cell subpopulation that is really eclipsed by the overall number of cells or the non-target cells in a sample. So our approach, instead of using fluorescently labeled antibodies, we use magnetically labeled antibodies. And our magnetic particles here are really small ones. And the idea was that if we could stoichiometrically label cells with these antibody functionalized particles, then if we could count up the levels of magnetic particles on the surface of the cell, that would give us a way to then quantitate protein expression, ideally at the single cell level, through magnetic label counting. And so this was something that we thought would have a lot of advantages. It would allow us to take really complex samples, hone in on a protein of interest, and then be able to do this type of quantitation even in the presence of a large background of non-target cells. But there was also a challenge that we needed to address, just related to the fact that the magnetic nanoparticles that we envisioned using don't give us a lot of magnetic force to capture the cells with. And so we needed to get smart about the microfluidics that we used in order to be able to pull these cells out of complex mixtures, and then to be able to profile levels of magnetic particles on the surface of these cells. And so the approach that we came up with is shown here. So here's our magnetic nanoparticles that are going to go into a sample, label a rare cell, leave the other cells alone. And then we have this microfluidic device that has a channel with a varying depth. And that varying depth gives us a linear velocity gradient, meaning that the solution is running through the device with a high velocity in the early zones of the device. And then as the channel is getting deeper, the solution actually slows down. We also try to counter the drag force on the cells by introducing these microfabricated X-shaped structures. And in doing so, we create pockets of low flow, and it makes it a little bit easier for us to capture these magnetically labeled cells in this device. And so what happens because of this velocity gradient and because of these X-shaped structures, cells that have high levels of magnetic particle loading stop in the early zones of the device, and cells that have low levels of magnetic loading then keep traveling, but eventually settle in the later zones of the device. The way this device is designed, there's about 100 different capture zones within it. And so we can kind of array cells according to their protein expression. And then to visualize them, we use immunofluorescence. So we come in with a cocktail of fluorescently labeled antibodies so that we can see the cells in the device. We can also layer in other markers above and beyond the one that we're looking at with our magnetic particle. And then we can create this histogram where we're looking at the single cell level at levels of protein expression. And then we can calculate expression indices for individual cells. We can take an aggregate expression index. But again, we can do this for a target subpopulation of cells and not have a large background of non-target cells interfere. And just to show you some data demonstrating how this works. So here's a few cultured cancer cell lines. We're characterizing levels of the protein EpCAM using flow cytometry in this inset. You can see that the 3 different cell types have 3 different levels of EpCAM. We can take 100 cells, put those through our device and get very similar profiles. And we've done the quantitation to show that it maps on to flow cytometry. And we can also split samples, introduce different nanoparticles with different antibodies, and then start to build up multi-marker profiles that way. And again, the power of this is that if we have, let's say, a whole blood sample, again, with billions of non-target blood cells, and we spike in a very low level of a cancer cell, then we can get a very reproducible protein expression profile, irrespective of the cell number. And so this first kind of configuration of the assay is one that looks at the expression of surface proteins. Over the years, we've also learned how to get at interesting targets inside of the cell. Here, of course, we have to fix and permeabilize the cells. But in doing that, we can look at things like messenger RNA expression, and we can also look at the expression of intracellular proteins. And so this system was really the first to be able to allow the isolation of rare cells and the profiling of rare cells using messenger RNA as a target. We were able to look at things like the androgen splice variant that's important for the management of prostate cancer by pulling prostate tumor cells out of the blood using that mRNA sequence. And as an example of an important intracellular protein, we were able to look at mice harboring xenografts of different pancreatic tumors, and we were able to evaluate whether or not the cells that were in circulation carried mutated BRCA2, which is an important gene to be able to track for the use of PARP inhibitors. So we've been able to develop these different approaches that allow us to, again, hone in on particular phenotypes, even when they're present at very low abundance. We're now moving to looking at other biochemical phenotypes, secretion phenotypes, other things that, again, would allow you to get at phenotypes of interest. And so now I'll just move over to a version of this technology that allows us to basically do the same thing, but using a non-destructive format, and in a format that allows us to recover the cells that we've profiled. In the version of the device that I just showed you, it's really an analytical measurement that we're making in terms of protein expression, but it's a little bit hard to take the cells out of that device for downstream processing. And so here, again, we're going to be using nanoparticle labeling and then sorting cells based on levels of magnetic loading. But here we're going to do this based on this idea of prismatic deflection rather than capturing in that device that I showed you with variable channel depth. And so the way that we do this is in the device shown here. So this is, again, a microfluidic channel. And the features in this channel are actually just guides that are made out of a very magnetic material. It's called meth glass. And the cells basically travel along these guides as long as they can. So they're traveling along the path, attracted by this magnetic material. But then as the guides start to turn, we introduce these angles into them so that they're turning. And at some point, the drag force on those cells will overwhelm that magnetic interaction. And then the cells will drop off of the guides. And they drop off into fractions that we collect. And we've been able to show that these fractions then harbor different levels of protein expression. And so just to show you some cells flowing through this device, you can see they're kind of sticking to the guides and making their way through the device. This one on the left-hand side, you can see the guides are just going straight. But then when we go over to a part of the device where the guides are starting to turn, you can see the cells dropping off of the guides and into these fractions that we can then collect and walk away with. So we've applied this to a number of different problems over the years, but the one that I want to focus on today, just as a really short story to tell you, is on combining this with a type of genome-wide CRISPR-Cas9 screening that Jason just spoke to you about, but really using this technology to focus in on particular phenotypes that may be present at a really low level during a screen. And so in this type of genome-wide screening, usually it's a live dead output that you're looking at. So did the cells live or die under a certain type of stress or a certain set of conditions? There's another interesting level of information, though, that's connected to modulation of protein expression or protein modification. But this is difficult to get at because what you would basically need to do is to take an entire library of CRISPR edited cells and put them through, for example, a fax machine to sort them, which is a pretty long process. There's significant sample loss with fax, and so it's not something that has been widely adopted as a way to do phenotypic screens. Our magnetic ranking cytometry approach allows us to sort through about 1 billion cells an hour, and so the level of throughput that we can achieve is a really good match with these types of phenotypic screens. And so again, the difference in throughput comes from the fact just that FACS is putting these cells into a single file arrangement so that it can look at levels of fluorescence and then decide whether to sort a cell or not. We're sending our cells down a super highway of these magnetic guides, and then we're able to get much, much more throughput. And so, as I mentioned, this is a joint effort with Jason Moffat, but also my colleague Stéphane Auger, who's also in the faculty of pharmacy. And so we decided to give this a shot and see if phenotypic screening using this type of approach could really help us accelerate the use of phenotypic screens. And so again, we'll have a library of CRISPR'd cells where there's a single gene knockout in every cell. And then we're sorting the cells according to differences in expression level to try to identify cells that have upregulated or downregulated or done something else to a protein of interest. And so the proof of concept study that we did just to try this out and to see how our microfluidic system would behave was focused on looking at a protein called CD47, which is considered a don't eat me signal. So this is a protein that is upregulated very often on the surface of tumor cells. And these cells upregulate CD47 to interfere with the interaction with SIRP-alpha, which is a protein that is expressed on the surface of macrophages. And the macrophages are basically checking for this interaction. And if they find it, they'll leave the tumor cells alone. So you can see why then the presence of CD47 would be beneficial. But CD47, you can certainly come at it with an antibody if you were thinking about drugging this as a target. But we thought maybe there would be other targets we could go after that would be modulators of CD47 expression. And maybe this would be something that we could get at through this type of phenotypic screen. So again, we're taking our CRISPR cells, we're using our microfluidic platform, magnetic ranking cytometry, to divide the cells into different subpopulations according to CD47 expression. And so we did this, and given that it was the first screen that we had done using this approach, we benchmarked against FACS, we benchmarked against MAX, which is another magnetic technology, but is not really that good at providing high levels of purity or high yields of cells. And so we were able to really establish that this was a better way to go after a phenotypic screen. And then in evaluating the hits that came out of the screen, what we identified as the top hit of the screen was a really interesting protein referred to as QPCTL. And basically, what we could learn about or find out about the function of QPCTL was that biologically it converts glutamine to pyroglutamic acid. And at the time that we did this screen, as we were punching in QPCTL and CD47 into all different types of genetic databases and search engines, we basically didn't come up with anything. There was no known association between these two proteins. And we honestly, scratched our heads for a while trying to figure out what the link was. But once we looked at the crystal structure of CD47, it became obvious pretty quickly what was going on. So if you look closely at the N-terminus of CD47, it has a pyroglutamate at the N-terminus. In this molecular cell paper where this structure was described, the authors commented on this and said, it's most likely a spontaneous modification that happens. But given that we had QPCTL as a top hit, what we realized is that what we had ended up screening for, rather than an actual change in expression, we were watching the antibody on our particle lose affinity for CD47 because pyroglutamate was no longer present on the N-terminus. And so that's how we actually turned up this hit. But it still turned out to be very relevant, because if you knock down QPCTL, you can see that the levels of modified CD47 go down, and that pyroglutamate modification on CD47 is actually essential for the interaction with SIRP alpha. So by interfering with the modification of CD47, you can turn off the interaction with macrophages. And so this was a puzzle that was a lot of fun to sort out, and turned up what we think is a pretty interesting therapeutic target. It turns out that the findings that we walked away with are pretty robust, and there were actually 2 other groups on 2 other continents that turned up exactly the same hit. So there was a group in Europe and another in China that had done very similar screens at the same exact time that we were working on this. And so there was a flurry of activity getting these papers out, but very nice to see this result and this important hit being replicated in other laboratories. So I'll just say a couple words in closing about where we're going with this. So this technology is incredibly scalable. So it's a microfluidic device. You can set up many of these devices in parallel. And what that allows you to do is large-scale screens, looking at many different cell types, targets, time points, and really being able to build lots of multidimensionality into this type of phenotypic screening approach. And so we have several programs that are ongoing, looking at proteins that are important immunotherapy targets, markers of stemness and resistance, also regulators of undruggable proteins. But another thing that I think is turning out to be a really productive direction comes back to this idea of interesting rare cell types. And another way that we're applying this and now getting some of our first results is for in vivo screening. So can we take a collection of CRISPR edited cells, implant them into an animal, and then pull cells out that have broken off from a tumor and are circulating tumor cells, or looking at tumor reactive T cells in animal tumors, and then being able to uncover important regulators of metastasis, and also of the immune response to tumor progression. So lots of interesting data sets emerging in these areas, and this is something that we're quite excited about moving forward with. So with that, I want to thank everyone in my group. These types of projects are large-scale team efforts for sure. I'll just mention in particular Mala Pudine, who developed some of those first devices with the 100 zones that I showed at the beginning. Peter Aldrich developed our prismatic deflection devices that really got us into this area of doing non-destructive profiling. David Philpott has worked with Peter on that effort, and they worked together on that CD47 screen that I mentioned, along with Barbara Mayer from the Moffat group. Mahmoud Labib has also done a lot of the work on developing assays for intracellular proteins and mRNAs, and has done some really heroic work. I thank them and everyone else involved, also our financial supporters, and you for your attention. So with that, I will ask the other panelists to come on back. And I'll ask everyone who is online with us to feed us some questions and help us out with the discussion here. And I'll encourage people that are just thinking about questions to ask or topics to raise that, you know, we've covered some really broad areas here: synthetic biology, you know, really novel approaches to tissue engineering and understanding the composition of tissues. You know, some cutting edge techniques on Jason's side in functional genomics. And so these are big areas that I think are all frontier areas. And if you have questions about where these are going, don't hesitate to just ask a more general question rather than something related to a particular experiment or a particular study. So I'll ask our audience to fire away with their questions. But we did get a couple kind of in advance for Hana, so we'll start with these. So the first one is, how easy is it to use different signals to activate the Degron switch?
So I'm not sure I understand the question. Is the question, can we use different keys? Or are there different types of switches? Maybe I should answer both. So actually, one of the things that I love the most about these systems is that they're very specific. They only activate with their key. And actually, our paper has a different Degron switch and key pairs that are in the same cells. So there are two pairs, designed pairs, and they don't crosstalk. And I think for synthetic biology, so one key cannot activate the other switch. They have to be paired up with their own switch. And for me, that's actually a feature, not a bug. But maybe there are applications where you want them to crosstalk. Now, we are actually vetting as we speak a phosphorylation-based switch. So maybe that answers the other side of this question. Can you design things that are activated by other than a peptide, by a key that is a peptide? And then the answer is yes. So I think I covered both bases, but if not, please feel free to, whoever asked that question, please feel free to follow up.
Yeah, so that's from Krishna, who is one of our PRiME faculty members. So Krishna, you can.
Okay, Krishna, thank you for the question.
If you have a follow-up question, just let us know. So another one for you: does the switch motif used in the locker protein need to have a natural tendency to form alpha helices? So what types of motifs have been tried?
So for the Degron, for the locker system, it's mostly alpha helices, but of course, David has a bunch of other things that use all kinds of folds. So, so we have other work with him on homodimerizing systems that use different folds and so on. So, but for the switch, these alpha helices just worked really well.
Great. Okay, Jason, we have a question here from Stefan. He's asking, how does the identification of autophagy pathway modulating TCL killing interfaces with the studies by Kelley and Mukherjee looking at calorie restriction and cancer growth synergy with IO drugs?
Okay.
So, I don't know if you can read the question. It might be easier to read it.
I know. I just read it. Thanks, Stefan, for the question. So I guess there are many paths we could take to answer this question, but one, I guess, simple way to start to answer it is just to say that many of the hits that we find in simple in vitro CTL killing assays are related to metabolic pathways that would be impacted by changing your media conditions or restricting, for example, glucose, right? So I'm not sure. And so then I don't know what the mechanisms would be related to that. For example, like if, restricting a metabolite in the culture would lead to something like PD-L1 downregulation. I don't know. We haven't looked at that, but there are a bunch of hits related to metabolic pathways that certainly would be influenced by caloric restriction that may impact CTL killing. Well, they do impact CTL killing. We just don't know what the mechanism is. So I don't know if you want to say anything else, Stefan, but
Yeah.
that kind of gets to your answer.
All right. Well, Stefan, chime in again if there was not a coverage of that. Oh, he's messaging me saying I can't. You can type another question into the Q&A if you want. Okay. So while we wait for some other questions to come in, I have lots. Zev, I had one for you just about the connection of what you're talking about to thinking about how to produce designer organoids as model systems. So how much have you guys done there to expand those types, using those types of effects?
I think it's a really great question. And the project really begins with our efforts to do just that. So early on in my time at UCSF, we developed a whole series of different tools for preparing organoids from dissociated cells with very defined shapes and compositions and three-dimensional structures and all this stuff. What became very apparent from those efforts was that no matter what we did, the cells would kind of go about their business and begin rearranging and achieve some steady state. And I think it was very naive of us to take that approach at the beginning, but it led us to begin to think much more about the dynamic properties and the physical properties of these cells. So returning to those earlier studies now, I think what we've learned is that we sort of begun to rethink about what's the role of microenvironment and the role of cell-cell interactions in determining what those measurable physical parameters are, right? So I gave a couple examples of things that we could change. One was cell adhesion. And there are, of course, many pathways that feed into different flavors of cell adhesion. Think of TGF-beta and EMT. Similarly, another perturbation was AKT, right? So you think of all types of different growth factors that are going to feed into the PI3 kinase AKT pathway. And so an interesting question would be, could we figure out a mapping between what some of those paracrine signals / microenvironmental signals are and the physical properties of the cells that sort of tip the tissues into one state or another? And that's a direction we'd be really excited to pursue. And we're starting to think about that a little bit. Did that answer your question?
Yeah, definitely. Yeah, lots to do there.
I have a question at some point. I can wait.
Go ahead.
Me too.
I have a question for both Zev and Hana, though. So it's a dual question. Okay, so, and this stems from something that Hana said at the end of her talk, where you said that you thought disease could just be a slight perturbation, right? Like just a tiny little perturbation. And I think, Zev, what you showed in terms of like these sort of broader distributions that shift just a little bit, but you get a lot of heterogeneity in what's happening before, or I guess even during steady state. Do you guys believe that? Do you think that most diseases are just like small tweaks that end up causing havoc over many generations?
Zev, what do you believe?
What's about
And cancer.
I think this is a really fundamental question, right? And I think what I'm gonna guess what Hana would say and throw it out there, and she can say that I'm full of it.
And I'll correct you.
You guys don't have to agree.
What I would have to say is that a lot of these systems have evolved a lot of redundancies. And there are many perturbations which have no effect because there's so much feedback, not just at the biochemical level, but also at the level of cell-cell interactions, at the physical level in terms of tissue structure, reinforcing different aspects of biology. But there's a subset of perturbations that are likely different in different tissues that can have a cascading effect on the system over time. So, in my presentation, I gave example of one, PI3 kinase, right? So It's a very frequently activated oncogene. It has a very specific and acute effect on these luminal epithelial cells. That disrupts tissue architecture in addition to all the other crazy stuff that PI3 kinase does, right? So there might be a dual effect of perturbations like activating PI3 kinase in the mammary gland, in that it causes survival, causes proliferation, but it also messes up the mechanics of the tissue and affects activity and all these other things. And it's likely that these perturbations are going to be different in different contexts. So that would be my 2 cents. So
I kind of agree with that, but I'm going to add, maybe 1 or 2 dimensions to this. So the first is that not all perturbations are created equal. You can, because of feedback and all of these redundancies, you can kill a lot of things in a cell and it just doesn't blink, right? But then it has Achilles heels. And these, my hypothesis is that a lot of these are involved in feedback mechanisms themselves, because that's the sensitivity you get by having feedback, right? So if you're lucky, all of these mutations and whatever hit things that, that the feedback can take care of. But if the feedback itself is hit, that's a problem. Now, is that most diseases? Is that some diseases? Does that ever happen? Never happen? I really don't know, but we haven't thought about disease in this way. The other thing is, and here's a fun thought about, how things can break catastrophically. And that happens in technological systems, that happens in the power grid, for example. There are all of these perturbations that you do that, on their own don't do anything. And then you get unlucky. And there are these small perturbations to different components just to align just right. And then the whole system breaks. So on their own, they're small perturbations. You do them individually, nothing happens, right? But they need to align in a certain way. And when you don't pay attention to them because they're on their own, they're kind of benign, right? And a lot of systems fail this way. Does biology fail this way? I really don't know. But I think there's a strong possibility that there are some failures happen this way. I don't know how to study that though.
Yeah, I think you could say, in drug development, you would call that synthetic lethality. Right? You have these 2 compensatory events, and if you hit both of them, then the whole thing blows up.
I mean, one important point, Hana, that you made at the beginning of your presentation that I think gets at Jason's question really nicely is that you actually have to think across scales to understand the effect of these perturbations. So a perturbation at one length scale can affect feedback at another length scale. And unless you realize that, you might not realize that you're really messing things up. So, again, an example being activation of a pathway in an epithelial cell, which affects cytokines that signal to the immune system, right? I mean, that's operating on 2 very different scales, and that might be missed in a simple assay.
That's right. Absolutely.
Shana, there's a question.
So we didn't answer your question, Jason. I'm sorry.
Yes, it did. Good. Excellent.
Shana, there are a couple of questions in the chat if you want me to.
Yeah, let me get to those. All right, this one was for me from Jamie. Is it possible to do 2 or more rounds of magnetic ranking cytometry in tandem using magnetic nanoparticles linked to different antibodies for further segregation of phenotypes? Yes, it is. We've done 2-dimensional sorts where you kind of go in one dimension and then you change out your sorting handle and then you go in another dimension. You can keep sub-segmenting subpopulations until you have so many you don't know what to do with them, but we can certainly do that. And he's saying, I'm imagining cells flowing in a microfluidic channel along one axis for the first separation and then onto an orthogonal. That's exactly how we do it. Is the low abundance of these cells the limiting factor in this case? Yeah, and that's a good question because, if you really want a profile, I mean, you can always get single cell level expression indices, but sometimes you really want to see the profile and really want to see, okay, how many cells with this phenotype do I have? And if you start subsegmenting too much, you lose the ensemble behavior in a way that can be a little bit confusing. So it always depends on what question you're trying to ask as to to whether you want to kind of keep them together as one population, do the analysis there, or start teasing them apart. It's a great question. Okay, so this is from Chad Altobelli for Zev. Do you think we can use a therapeutic to alter the properties of the basement membrane and enhance the relative adhesion of LEPs and MEPs?
Yeah, I think it's another interesting idea. You know, there's been a hypothesis in the breast cancer field for many years that the myoepithelial cells represent this sort of critical tumor barrier. And it turns out, I didn't mention this, but they're actually the cells that are responsible for making the basement membrane. What's not clear to me is like, there's a fair amount of redundancy in terms of the specific integrins that cells express and the ECM proteins that they bind to. And the basement membrane is a very complex material. I think the answer to your question is yes. How to do that, I have no idea. So, you know, it's something that, you know, we should think about. I think it's a really good question.
So Zev, could you just to follow that up, can you grow these on scale and reproducibility that we can just do a phenotypic screen for it?
Yeah, I mean, wow, that would be super cool.
I guess that means no, not yet.
Well, you know, it's not clear, right? So, you know, I think a major goal that we have as a group would be to be able to do the types of studies that Jason described, but on whole tissues, where the information that we got out would report across 2 different length scales. So the scale of individual cells and how they behave to the perturbations, as well as the coupling between cells. And in order to do that type of screen, an assumption that we have, which may be completely false, is that we need to get to more homogeneous assays in terms of the organization of tissues. So it's getting at exactly the point that you're raising, Michelle. And so,
Yeah.
you know, could we use 3D printers to do this? This is something that, you know, people are beginning to do and reporting more reproducible results. We could use some of the techniques we've developed in a lab. That's sort of the goal, right? So we're working towards that.
Yeah,
But
I
can
think we
you
should
give us,
talk about it.
sorry, yeah, I actually want to know what is the order of magnitude of the heterogeneity problem? Like if you were to repeat this thing for like 100 times, you would get 100 different structures?
So
What is the scale of the heterogeneity?
Well, it depends on what you're quantifying, right? And in our case, we chose something very simple to quantify because we could, but happens to be relevant in the system. And you know, the distributions are quite reproducible. There's systematic error. So, you know, sometimes the sorting of the cells, we sort a cell out as being luminal and it ends up being myoepithelial, and we have assays to test for that. But if, you know, if we really carefully control things, you know, we get basically a similar distribution every time.
Yeah, you know, because I think, you know, we and others do some pretty subtle phenotypes now. I don't think you should let heterogeneity a priori dissuade you. You know, if you can grow them on enough scale, then we can just look at distributions and shifts in
I
distribution.
mean, think about heterogeneity as a feature, not a bug. It's a natural perturbation.
That's an excellent point. What we often see is that in the screened state, when you have a hit or a disease, the distribution narrows, that's a measurable parameter. Things get more similar.
I mean, that would be cool because, you know, the pipeline for analysis is in place. So yeah, we should definitely talk about that, Michelle. That'll be fun. Can I ask a question? I have a question first for Jason, and then for Shana. Jason, you showed a slide about 3/4 of the way through the presentation where you were, I think you were clustering by phenotype. Is that correct? It was like a heat map.
That was yeah, that was CTL killing phenotype. Yeah. So
was each cluster, did that represent a distinct mechanism of escape from CTL killing?
Either escape or resistance or sensitivity. So we had both ends of the distribution in that clustergram, and we clustered the tails. So we derive a profile for each gene and then just do pairwise clustering. Mm-hmm. So each set of branches in the tree is really a bunch of genes that behave similarly across all screens and time points. So we screened 6 different models we actually have another 2 and then multiple time points along the way to get. What usually happens though in these killing screens, it's just like a drug screen, you end up selecting for resistance hits the further out you go if you keep on adding drug pressure. So earlier on, we tend to get more sensitizing hits than resistance hits, and later on we get more resistance than sensitizing. So we don't know where the sweet spot is all the time with each different model. So we tend to do multiple time points and then figure out what the best representation is. Yeah.
What's interesting there is it suggests that there's a pretty narrow number of ways that these cells can escape the killing. So have you turned the experiment around and taken subsets of those mechanisms and done drug screens to see if you can compensate for them?
Yeah, so some of the, like there were a couple screens in there where, for example, FIT-M2 was the strongest sensitizer to interferon gamma signal, which was an interesting hit. But then we made a FIT-M2 knockout and screened it for interferon gamma sensitivity, or sorry, resistance, right? So you don't get many things, but you do get a few things. So we did turn that around, and those were those sunbeam plots that I sort of showed and maybe went through a little bit too fast. But we do have genetic interactions now where we think we can kind of rewire things a little bit to resensitize it, say, well, that you're thinking about the opposite end. So if you find a resistance hit like JAK or STAT, can you resensitize those guys? Or
reprogram the immune cells specifically.
Yeah, so the immune cells, we've only just started to begin to play around with. Other people are doing that. Like Alexander Marston at UCSF is a good example of someone who's been really probing the T cell side of things, right? It's funny, a lot of the pathways are similar from the T cell side and the cancer intrinsic side. So there's a lot of crosstalk between these cell types, and other immune cell types like NK cells also use very similar mechanisms to kill tumor cells than activated T cells do. So yeah, it's a great question. We're kind of exploring that now.
And if I could ask the second question, Shana, I was just thinking about the geometry of the separation channels in your devices. And is there any theoretical limit to the sensitivity of separation in that system? Like if you make the channel longer and longer and longer, do you just get better and better separation?
Yeah, absolutely. I mean, if you just keep going, you give yourself more capture zones. And I'm not sure what the limit of fabrication is. That's probably what you would eventually hit. You know, you just can't find a substrate that's going to give you like a 6-foot channel. And then also the throughput, you know, the more channel, the more time it's going to take to process. We find with 100 zones, that's good for most things. If you're really looking at like wildly different levels of expression, you'd almost be better off kind of doing a high, low, medium, and then really spreading them out so that you get the really distinct subpopulations, but you can do it either way.
And
can I ask a really stupid question, actually? Really stupid. Can you make the cells gobble up the magnetic particles?
Yeah, that's a great question.
To label like internal states?
Yeah, so we have a proliferation screen that we've done very recently where we let the cells eat magnetic nanoparticles, and then we can look at how many particles they've engulfed, and then we can also let them keep dividing, and then you kind of wash the particles out as they divide, and then you can find the cells that are most active in proliferation because they've gotten rid of all the particles that they ate. So that's actually a really cool phenotype to be able to latch onto to get some biologically interesting information.
Sorry, Zev, I interrupted you. Did you have another question? I really wanted to know if the cells can eat them.
Yeah, I just had sort of a follow-up question, which was, you know, just in terms of a quantitative comparison of the sensitivity between and the dynamic range of the FACS and the magnetic-based separation systems. Have you thought a little bit about how far you could push it in terms of, you know, fine-grained differences in detection?
Yeah, I mean, it really depends on the cell type, what you're trying to separate. There's some things that FACS is just not a good option. Like, you know, if you want to go into a tumor and find the T cells in the tumor, like you don't want to go anywhere near a FACS machine, right? You're never going to get most of the cells back, and you're probably also going to get banned from your FACS facility for doing that. So, you know, I think it's those types of really challenging problems where we're better equipped just because of the size of the channel and all the processing power. For really, you know, small changes and also multi-label type, you know, multi-protein type analyses were challenged. And we have not challenged resolution as much as sensitivity. And for sensitivity, we're a lot better, like with sticky cells, we're a lot better. Cancer cells were a lot better than FACS. I mean, FACS and flow cytometry were really developed for immune cells, right? So most immune cell types are fine until you start making mixtures, right? And you're starting to mix immune cells and cancer cells, and then you end up with problems. So again, there's like 100 different answers to that question, depending on like the cell type, the marker, exactly what you're going after. Okay, any other questions from our panel? This has been a great discussion.
I have one last question for Jason. Sorry, somebody else.
No.
Somebody else wanted.
No, Hannah, go ahead.
Yeah.
I was going to do more of a wrap up.
Since it's on everybody's mind, like machine learning and AI and so on, it's all over the news. Are you incorporating any of that in your workflows?
Yeah, that's a good question. So we have for sure, but I think, I can't remember who said this, maybe it was yesterday, we still need to do experiments, right? There's not enough information.
Yeah, I think so.
I'll take credit for saying that.
Okay, Michelle, yeah.
But yeah, the answer is yes. Like, for example, we developed this convolutional neural net for deriving Cas12 guide designs for that chimera system that I mentioned that we just published, and that works really well. I don't think we're there yet for example, genetic interactions, or even inferring genotypes that might be important for context-dependent sensitivities. So that I think we're still a long ways away from that. In my opinion,
this is because of the data, or because the methods, the AI methods, don't exist?
I don't think we have enough data yet, but that's just my own bias. Maybe somebody who's really smart with AI will figure it out before we have enough data from our lab or from. But it's a good question. I don't like, how do you infer genetic interactions with AI? I guess there's the yeast data, it's a great data set, but then you have the problem of mapping all the functional orthologs over and stuff. So
yeah, I mean, there are people actually using really clever kind of concepts from metric learning and natural language processing to do such things, but it's really just being worked on right now. And I just wonder, how far is that going to get us, and what kind of data is needed for that to be even useful?
That's a good, maybe this is a good intersection for a student or someone to come in and start thinking about.
Yeah.
Good job, Jason. That was going to be my question.
Yeah.
So all of you collaborate broadly, develop technologies, and use technologies to develop new biology. So how do you choose collaborations? And towards the goals of PRiME here, how would you see these connections being made and going forward? Anything spark while you were talking or listening?
I mean, all of the systems that were discussed today, there is just a lot of interesting models there. I mean, yesterday we talked about kind of cracking open biology to expose new, weird, and wonderful targets that can be very interesting to characterize just because of the science and also potentially therapeutically relevant. And I think everything we heard today is a way to like crack the nut, right? And then we have the folks on the other side that can do the biophysical characterization and the biochemical characterization. And so I think we have a nice interface between the technology development and then the biological characterization. So no shortage of material here, that is certain.
I do have a question about the approach that you guys are envisioning. So there are two ways to go about, things in a big enterprise. One is to say, okay, we're just going to have everything from the ground up, and whatever just kind of bubbles up, that's good enough. Or just focus on, say, okay, we have these 2 or 3 strategic priorities, and who wants to take a stab at this? And I'm just wondering which approach you want to take. Because I think that defines kind of how one can go about catalyzing interactions, right?
Yeah, I mean, Shane will have a more holistic answer to this because she runs the whole program. But for me, when we were talking about what to do for this kickoff symposium, it's which 2 of the 10 or 12 things that popped into our heads immediately that both universities have strengths in, what's the best way to overlap, and then in those, how to start picking speakers. So it's really, I guess in that sense, there's some grassroots, but then there's some sort of random decisions you make based on who you know. So I think what is always interesting in this kind of thing is how to go from those networks to linking to networks you don't currently know. And then what's the best way to do that? So how can we take the people who are on the calls in the last 2 days and say, oh, I know somebody in this totally other area. I'm going to pull them in and see if they'd be interested in working with somebody in this other area.
Yeah, and I do think there's room for both. I mean, we're making the rules here in terms of how we do this, and I think we can do some kind of top down. I mean, there are people involved in these sessions that have kind of always wanted to really get a collaboration going in this particular area, and so the interest is already there. But what I'm hoping, I mean, we've had great turnout both yesterday and today, and tons of trainees, and I would encourage them to just think creatively about everything that they've heard about. And if they can see a link to what somebody else is doing and what they're doing right now, propose a wild and crazy idea and, get their supervisor to buy in, obviously. But then, let's get new areas connected through the trainees, because in my experience, that's where the really new exciting stuff can come from, in addition to faculty to faculty type interactions. So I would encourage us to do both, and then we can see what works the best. But my prediction is that both mechanisms will work pretty well.
Okay. In that spirit, Jason, I'm going to throw you a curveball. So if you can tell me which gene I need to boost up or regulate or amplify or dampen with a circuit in order to make a super killer T cell, I can probably do that. So and the idea there is that, what we're using right now for like to put cars in are just your vanilla flavor T cell, but there's no reason to believe that this is the right platform. Actually, it could be that another platform will have certain things up, others down, and various other things modulated so that they're more malleable to the environment might be the good platform. So can one marry your high throughput kind of exploration of the capabilities of these cells with a design purpose?
I think,
and this is one idea. It just occurred to me right now as we're talking.
No, but I think like you and others are already doing that, right? Like, the Limb Lab and are already beginning to engineer.
We're not messing with the T cell circuits though. We're not messing with the internal mechanics. We're adding stuff on top.
You're adding stuff on top, but
we're not rewiring internal pathways really in any meaningful way.
But I think you're absolutely right. You can mess with the T cell circuitry. You can mess with the genetics of a T cell. I mean, this is where a lot of the allogeneic efforts are going, right? To mess with the genetics of T cells and making off-the-shelf T cells, but not just T cells, but other cell types like NK cells. And, you know, even there are companies that are making CAR macrophages now that are certainly going to be engineered more than just sticking a CAR in. So I think, yeah, no, this is for me, this is very exciting biology. I think there's a lot of fun academic science to do, but also potentially very useful.
Right. I mean, I pitched it as an applied thing, but in my mind, actually, my lab this whole applied stuff is very new to me. The way I actually started doing synthetic biology and cell engineering is at the service of actually interfacing with endogenous biological system, perturbing them in precise and meaningful ways, and then getting bigger and more clearer understanding of the systems themselves. So it's like synthetic biology at the service of, basic knowledge. And we did that in, very small scales. Others have done it too in small scales, but I don't think that vision has ever been even realized, where you meaningfully marry cell engineering and high throughput technologies and genomics and so on, to increase understanding.
Yeah, no, I think we're getting closer to being able to make much better predictions about how to do that too, right? Because a lot of the basic components you may want to engineer into like a CAR or some sort of therapeutic cell, that's moving at lightning speed. And then you've got these, very, high resolution screens that you can do on a academic side to really start to tease out not just single gene perturbations, but multiple genes at one time, just to get a better understanding what might be better to engineer. But yeah, no, there's a bridge there that's going to connect at some point, right?
Yeah, I think that would be a great topic for a follow-on event. I mean, we have a ton going on in Toronto around cell therapy, cellular engineering, and the intention here, and we should probably also wrap up because we're at 3:30, is that we're going to kind of cycle through all of our partners and then keep tabs of all of the kind of areas of synergy, and then maybe come back with a four-way event where we get folks from UCSF and NUS and ETH and UofT, and then come back together and maybe talking about using synthetic biology for cell therapy applications, this kind of thing, and the interface with functional genomics. That could be a really cool set of discussions around that. So I think we'll leave the discussion there. Thank you so much, everybody, for taking the time this afternoon. This was just a wonderful session. And I thought we couldn't top yesterday. I was a little worried we weren't even going to come close because it was such a fantastic session, but we did it. It was amazing. So thank you, everybody. The science was just wonderful, and appreciate you taking the time, sharing with us. Thanks for everybody who joined us as an attendee. Greatly appreciate you tuning in. And so, as I commented, this is just the first crack at getting our institutions connected. And so we'll kind of circle back after we've made the rounds and also see what's happening in the world and as to when maybe people can start moving around again, and then we'll take it from there.
Yes, and if you have any ideas, send them our way.
Yeah, absolutely.
Great. Thank you, everybody. Thanks so much to the speakers.
Bye-bye.
Take care.
Bye-bye. Great to see you all.
Bye.